So let me start with thanking John and Pramila and all the organizers. It's just been a terrific conference. All the talks today, as well as the earlier talks in the series, amazing. I also want to make sure to thank the individuals with autism, their families that participate in the work that's informed the thinking I'll share with you today. Obviously, our collaborator network aims to trials, Jamie from ABCCT, Autism CRC in Australia, and really the global set of consortia that are active in autism. Our study sites, patient advocacy groups that have given us lots of formal and informal feedback and guidance, and then of course key partners within Roche, including Kevin Sanders and Julian Tillman, who both spoke earlier in the conference. Disclosures: obviously I'm a full-time employee of Roche, and the views expressed in this talk are not necessarily those of Roche. So I want to start with some good news for 2020, which has been sometimes difficult to find. And the good news that I wanted to highlight here today is the reversal of a trend sometimes known as Irum's Law, which is the exponentially decreasing number of new medicines based on every $1 billion spent in R&D. So this kind of reversal of Moore's Law in the case of drug discovery had held over about 70 years up until the most recent and most comprehensive analysis of this historical data in the mid-teens. And what has been published just in the last few months by Ringel et al. In Nature Reviews Drug Discovery is new data since 2010 that shows that somehow we've broken that 70-year historical trend. So this is very encouraging, also somewhat surprising. And so what they've done is to go back and show, yes, indeed, the residuals here at the upper right strongly depart from the best fit line. What else might have been changing over the same interval that could be responsible for this reversal of a 70-year pattern? And there were three things that they identified in their paper that they thought could account for this. So one was a drastic increase over the same period of time in the number of new molecular entities with supporting genetic data. Alongside that, there was also an increase in the number of approvals taking place either explicitly for rare diseases or at a more granular level than the basic level in ICD-10. So all three of these together offer an interesting and maybe optimistic picture for what we can expect from R&D efforts in the future as pertains to getting new medicines for conditions with major outstanding unmet medical need. But the news is actually even a little bit better than that. So this analysis only accounts for data up until 2018. Where we have, of course, traditional small molecules, as well as some large molecules included. But really, there's been two major changes just in the past few years that give even further reason for optimism. One is that there's now an ever-expanding understanding of what the druggable genome actually means. That is, of all the protein-coding genes, there's actually only a tiny subset of those which existing treatments target. And in fact, of those that are not targeted, quite a high proportion of them, relatively speaking, appear druggable. That's for traditional small molecules, but on top of that, it just, again, actually in the last about 2 years, we've seen other neurodevelopmental conditions like spinal muscular atrophy have a whole array of new treatments brought to bear using entirely new technology, including AAVs, ASOs and LNAs, as well as now we believe small molecules may even be capable of RNA splicing. So all three of these modalities have been brought to individuals with spinal muscular atrophy just in the last 2 years, are now approved and on market. The question for us here is, next, as one of the most highly heritable psychiatric conditions, we could expect this new array of technologies and the increasing amount of success we expect to follow from having a clear target rationale involving human genetics to really revolutionize the treatment of autism. So the safe approach to this, and this is a graphic that's been shared, I think earlier today by Matthew, and then of course also shared by Dan Geschwind in his talk last Sunday. The safe approach is really to target these known rare forms of autism, right? So there we have a clearer causal chain. There are often advocacy groups set up specifically for those rare forms of autism. And I think it's great that we have such a rich collection of biotechs, both large and small, pursuing that area. But this, of course, leaves the majority of folks with autism without, really a very clear path here, with, I think, very notable exceptions, like much of the data that has been presented earlier today, very exciting. But we still need to acknowledge this is sort of the great unknown. So the question is, how do we actually fill in the map, right? We have a collection of rare conditions where we feel like we understand they're tractable using one of now a whole array of different therapeutic modalities we have at our disposal. But there's a broader section here actually making up probably the majority of individuals with autism where we really don't know, is this sort of one big thing? Is it a collection of many little things? Is it some sort of combination there? So one thing is clear. In order to fill in those sections of the map, there's going to be more data that's required. So what I'm sharing here is a figure from a power analysis presented in Nature Genetics by Zhang et al. 2018, where they use linkage disequilibrium and LD regression in a mixture modeling framework to try to predict exactly how many cases do you need from each of a whole variety of conditions in order to start chipping away substantially at the estimated heritability here on the y-axis. And so in blue, we can see the case for autism spectrum disorder in this context of a large number of other different conditions. And so our current estimates of where we're at, more about that on the right side of this slide, is that if one were to sum across all of the major cohorts, at least that we're aware of, that have genotyping data, then we can get right about here to this inflection point, which it should be mentioned is for a rather simple analysis technique of kind of univariate risk computation. But nonetheless, we think that it's actually possible to get to that point where the rate at which we can explain additional ASD risk is exponentially greater than it has been historically as soon as we can reach that sort of inflection point. This is going to be a huge effort to try to pool that sort of data. More on that in a moment, because pooling is probably not going to be the most scalable approach here. But it is clear we need more data that we can just eke out, at least for these simple analysis methods, enough mileage to get to that elbow in the curve for autism, if we're capable of pooling or combining across these data sets in some other way. And as we sort of embark on that attempt to fuse all of this data and better understand what, in fact, the majority of individuals with autism, where their risk is sort of coming from genetically, and use that understanding to leverage all these new therapeutic modalities against those targets. We are also going to need to be running trials where we don't have the hypothesis quite right yet, right? And that puts us in a more post hoc situation where what we need to be able to do is use the most powerful methods we can find to do the subgroup discovery from our own clinical trials. And so Kevin Sanders' talk from last Sunday actually talks a little bit about this in the context of Alavaptan for a worked example. And it is a case in point of the broader problem here. So what I'm showing you on this slide plots statistical power at a rather liberal alpha, 0.1, right? Given a number of different effect sizes in a responder subpopulation. So we could see a Cohen's d of 0.4 or 0.6, all the way up to 1.2, maybe in our wildest dreams. And then I have different lines plotted for the prevalence of that responder population. And the question that's being answered in terms of the y-axis is how often, if you did have an effect size of that amount and a responder subgroup of a given prevalence, how likely would you be to actually detect that effect in the group? And so what you can see is that, unsurprisingly, if everyone's responding in these yellow curves, right, we have a quite good chance of detecting that, even at sort of modest sample sizes, right? But once we start talking about 25% response, which I think may be a kind of upper limit at the moment for some of the more idiopathic forms of autism, then we really need absolutely massive, maybe even implausible effect sizes in order to be able to detect that at a reasonable sample size. So this is using the most powerful test that I can find for this kind of approach. It's an Anderson-Darling test. It's basically an extension of tests that would look for normality in a particular sample, and with added sensitivity towards discrepancies observed in the tails of the distribution. So, right, it's clear that we can go this way, but any results that we obtain from this sort of method are going to be, at best, hypotheses that need subsequent tests in that responder subgroup. And that's exactly where we are with Belabeptin, as Kevin sort of alluded to. So this is collectively what I call the joint identification problem. And maybe we can discuss whether that's a good name for it or not. But the basic point is that to break Irum's law in other areas, for example, borrowing on new successes in rare conditions where we do have genetic evidence, we of course need to fractionate autism into the set of many to possibly innumerable, modestly rare to possibly ultra rare conditions that most of us actually believe that spectrum to be. Now, in order to do that, to fractionate autism, we're going to need a better understanding of the phenotypes, of the causal or genetic risk factors, and improved markers at the intersection of those two. So what I'm going to spend the remainder of my talk describing is two possible solutions to this large problem. So one is to try to solve this joint identification problem directly in large sample data. So we're looking here for scalable approaches that can feed very data-hungry techniques. And critically will enable sort of genotype or phenotype informed recontact from those subject pools subsequently in order to confirm the tests that we expect or the hypotheses that we expect we will pose from our own post hoc biomarker-based stratification efforts from ongoing trials. There's a second approach as well, which I'll talk about at the tail end, which involves a renewed emphasis on biomarker modalities, which maybe don't scale quite so well just yet, but where the causal chain can still be made transparent back to these newly druggable targets, and could therefore more clearly inform target selection. So in general, I have this little graphic on the right about this chicken and egg problem, or chickens and egg problem, right? Where we can't really better understand the phenotypes entirely until we have a better idea of who belongs to which subgroup or another. Maybe we're not even entirely confident that that problem is solvable without better understanding the phenotypes. And so this is the sort of chicken and egg problem that the two solutions I'll describe in a moment are meant to help us address. So the first is, I'm really pleased to be able to describe this today, although there's an additional announcement I expect maybe impending in the next few weeks related to this general effort. It's called the Autism Sharing Initiative. And so you can go to autismsharinginitiative.org right now and find a rather simple web page at the moment. But the core idea behind the ASI, which was founded by Autism Speaks and DNA Stack, and which Aims to Trials has now joined, is instead of trying to reach these critical inflection points on the power curves I showed earlier by pooling data, which involves extremely laborious data sharing agreements, many of which may actually violate the underlying consents that the donors signed up with, and so it's just not really a workable approach, is rather than moving the data to the analysts, is to move the analysis to the data. So this is not a terribly new idea. It's just an infrastructurally challenging one to accomplish. And that's where I think this effort is particularly strong and will have legs, in part because it's been announced as a Global Alliance for Genomics and Health driver project. So what this means is that the Autism Sharing Initiative joins the ranks of other efforts like the All of Us program, like ClinGen, like Elixir, and in doing so can benefit from the underlying APIs, infrastructure, ethical and privacy frameworks that have already been put in place for these other efforts. So that's really an important ingredient here. And what we're looking for, of course, is additional consortia to join in this effort where existing data access committees retain their control over the data they have, but we agree on a common framework and a common infrastructure by which analysis can be moved to the data should any particular data owner or data generator approve the performance of that analysis on their data set. So I think we're extremely optimistic about the Autism Sharing Initiative, in part because of where we're at with the trials of today, where we know we are recruiting a heterogeneous population. We're imposing the kinds of selection criteria that we feel like we can already, but at the end of the day, we know that we have uncertain throughput into the trials, so recruitment is a huge challenge, and what comes out of the other end is itself a mixture, which, as powerful as that Anderson-Darling test is for uncovering departures from normality, for example, the presence of a mixture distribution, these are still just challenging computational problems. So in the future, we would hope to be able to use the Autism Sharing Initiative to actually selectively recontact those subjects that may harbor a particular biotype that we, through our post-hoc stratification analyses, believe may be associated with differential drug response. And being able to selectively recontact them and potentially canvass them for their interest in an RCT would be critical for addressing the challenge posed by really low prevalence subtypes, which we may well find make up the majority of this great unknown in ASD. So do we have to wait to really make progress until we have this infrastructure live? So I think there's a very clear answer there, which is no. And the reason has to do with other computational breakthroughs and different sorts of biomarker modalities that we can explore that may actually offer a different view onto the same joint identification problem. So what I wanted to highlight in that context is metabolomics. I think this is one example. Immunological markers like those spoken about by Judy are another great example. But the key thing I wanted to illustrate about metabolomic approaches that make it particularly suitable modality for this kind of problem is that if we have that great unknown in autism representing a combined influence of many hundreds or perhaps thousands of different genes, right, all of individually small effect, then we're always going to be really underpowered up until we get to the upper extremes of the power plot I showed you from Zhang et al. In 2018. But if instead we can actually measure something that reflects the cumulative contribution of many genes, then we can not only preserve the transparent mapping back to the genetic loci, which new therapeutic modalities will allow us to target very cleanly, but we may also be able to find sort of greater convergence or more highly prevalent subgroups, which then ease some of the challenges while we fully bring the Autism Sharing Initiative infrastructure up to speed. And one example of this I'm showing at the bottom right. So Jürgen will speak later this afternoon, and I'm sure share many more interesting ideas about how to approach this kind of topic. But if we think about having a large number of individuals, say on the y-axis of this matrix, and a large number of different biomarker features, say metabolomic and or genetic features on the horizontal axis here, of course, we can perform clustering on this matrix. Typically, that's done on one axis or one dimension of the matrix separately from the others. So we may try to find clusters of features that look different. We may find clusters of individuals that look different. But really, the problem that we're trying to solve here is to identify both jointly, right? Both the features that pertain to individual subsets of subjects. And there are techniques for this. Biclustering is one of several, but it is particularly data hungry. And so the key insight from this sort of metabolomic kind of approach, at least from the computational point of view, is not only that it represents this kind of cumulative effect that may be especially suitable for that great unknown in autism, but because we have some idea about what the causal chain must be, right, in large part, we can actually constrain the relationship among features in a clever way. So that if we know, for example, in this middle set, again, just a illustrative graphic, not real data here, that there were a set of genes or reactions that were altered in a subset, we could say with some confidence that there should also be alterations in some other subset. The converse may not hold, but that should, given the causal chains that we know and have been sort of laboriously documented, although incompletely, in the metabolome over the past several decades. So I think this is a kind of naturally complementary technique to the larger problem that we have with joint identification on the basis of genetics. So with that, I wanted to summarize what I've said, because I've realized that I've gone to a number of different modalities here and have also spoken in fairly theoretical terms. But there really is a plan for action here, and what we need is basically a lot of the pre-competitive consortia work that we'll talk about later in the session to congeal around some common frameworks for bringing the analysis to the data. So as those existing pre-competitive consortia continue to proceed with their sort of endophenotyping first strategies, which we should note have been actually quite successful in certain areas, right? So Jamie shared the really exciting data from ABCCT, which is very parallel to what's been observed in AIMS trials. And that is, I think, itself its own source of potentially transformative stratification biomarkers for clinical trials in autism. But there's a broader framework here too, right, of innovations in the therapeutic modalities we can bring to the picture. And in that respect, things like genomics and metabolomics do have a privileged position. They're closer to the metal, or closer to the biology in this case, in a way that upon the discovery of a stratification biomarker, it can much more directly instruct the therapeutic modality we would choose to pursue a particular subpopulation than something more endophenotypic could. So in order to solve this kind of joint identification problem of needing clearer phenotypes and needing that clearer causal path from the genetic evidence to the phenotype, along with scalable markers at the site of action as well as downstream, we are assembling the Autism Sharing Initiative together across industrial partners, academia, and patient advocacy groups to federate these sorts of large samples, enabling stratified recontact and the deployment of scalable markers. So I haven't spoken much about our efforts on the digital biomarker side here at the upper right. Julian in his talk earlier alluded to that a little bit in the Q&A session. But I think that's also only sort of half of the picture. It's a very important half, but there are also the computational methods that we would deploy, even on techniques that are maybe a little less scalable, but where some of the biology is already better documented in ways that let us constrain that joint identification solution. So with that, I'll conclude a little bit early and happy to take questions. Thank you.
Great. Thanks so much, Chris. You've unintentionally set up the rest of the day perfectly. Coming up after the break, we've got Diane Stevenson of the CPAP Institute talking about pre-competitive consortia and private-public partnerships and how we can work in this sort of model for autism moving forward, and Jürgen Hahn talking about machine learning and metabolomics, which is near and dear to us at BioRosa. Nicely done. So a couple questions have come up. One, the ASI sounds great in theory, but how do you incentivize industry partners to participate, given that IP generation may be something that limits their willingness? Just thoughts there.
It's a terrific question. I can understand certainly where the question comes from, and I can't speak for other groups, but it seems to me that we're plenty, and any company, large or small, interested in running clinical trials in autism has this problem. And if there were a framework in place that would allow us to take some of the post hoc hypotheses we expect we will have at the conclusion of any given trial and more tractably run that, the incentive is already there. And the discovery of the targets themselves is not typically seen as a key part of the intellectual property strategy, at least in my experience in the large kind of pharma and biotech world. It's really the chemical matter, right? And that if we protect that, we compete on the chemistry. The targets are something that actually we would love it if more people out there knew about the same targets and were working on them and could bring more science to the table, help us all find better chemistry in the future. So I do acknowledge the challenge, but it's just that practically speaking, if you're invested in working in autism, this is a problem you need to be solved, and you would rather be sort of at the table in helping to design a solution than outside it. So I think the problem is more apparent than real.
Cool. I couldn't agree more. Sounds like you've touched on this a little bit, but it did come up, just if you can speak to it more directly. Is the ASI initiative limited to industry only, or is it also open to academia? And who is funding this on an ongoing basis?
Yeah, it is absolutely open to all. And so we've been in discussions with a number of different academic partners, as well as some additional industrial partners. That's, there will be more details around that, I hope, in the next few weeks, maybe even sooner than that. So yeah, that was one aspect of the question. Can you remind me the other?
So yeah, so the other aspect was how's it being funded?
Oh, yeah, right. So, I mean, the Global Alliance for Genomics and Health does offer support in terms of the development of the infrastructure and the engineering talent that goes into implementing that for any given driver project. But it is this further imminent announcement that is going to be relevant to the funding needed to really build out the system. So, yeah, more to come on that.